Strategies for enhancement in food production. With ever increasing population of the world, enhancement of food production is a major necessity. Biological principles as applied to animal husbandry and plant breeding have a major role in our effort to increase food production. Several new techniques like embryo transfer technology and tissue culture techniques are going to play a pivotal role in further enhancing food production. Animal husbandry. Animal husbandry is the agricultural practice of breeding and raising livestock. As such, it is a vital skill for farmer and is as much science as it is art. Animal husbandry deals with the care and breeding of livestock like buffaloes, cows, pig, horses, cattle, sheep, camel, goats, etc. that are useful to humans. Extended it includes poultry farming and fisheries. Fisheries include rearing, catching, selling, etc. of fish, mollusks, shellfish, and crustaceans, prawn, crabs, etc. Since time immemorial, animals like bees, silkworm, prawns, crabs, fishes, birds, pigs, cattle, sheep, and camel have been used by humans for products like milk, egg, meat, wool, silk, honey, etc. It is estimated that more than 75% of the world livestock population is in India and China. However, it is surprising to note that the contribution to the world farm produce is only 25%. That is, the productivity per unit is very low. Hence, in addition to conventional practice of animal breeding and care, newer technologies also have to be applied to achieve improvement in quality and productivity. Management of farm and farm animals. A professional approach to what have been a traditional practice of farm management gives the much needed boost to our food production. Let us discuss some of the management procedures employed in various animal farm systems. Dairy farm management. Dairy is the management of animal for milk and its product for human consumption. Can you list the animals that you would expect to find in a dairy? What are different kinds of products that can be made with milk from a dairy farm? In dairy farm management, we deal with processes and systems that increase yield and improve quality of milk. Milk yield is primarily dependent on the quality of breeds in the farm. Selection of good breeds having high yielding potential under the climatic condition of the area, combined with resistance to disease, is very important. For the yield potential to be realized, the cattle have to be well looked after. They have to be housed well, should have adequate water and be maintained disease free. The feeding of cattle should be carried out in a scientific manner with special emphasis on the quality and quantity of fodder. Besides stringent cleanliness and hygiene, both of the cattle and the handlers are of paramount importance while milking, storage and transport of milk and its product. Nowadays, of course, much of these processes have become mechanized, which reduce chance of direct contact of the product of the produce with the handler. Ensuring this stringent measure would, of course, require regular inspection with proper record keeping. It would also help to identify and rectify the problems as early as possible. Regular visit by a veterinary doctor would be mandatory. You would probably find it interesting if you were to prepare a questionnaire on diverse aspect of dairy keeping and then follow it up with a visit to a dairy farm in your locality and seek answer to the questions. Poultry farm management. Poultry is the class of domesticated fowl birds used for food or for their eggs. They typically include chicken and duck and sometimes turkey and these. The word poultry is often used to refer to the meat of only these birds, but in a more general sense, it may refer to the meat of other birds too. As in dairy farming, selection of disease-free and suitable breeds, proper and safe farm conditions, proper feed and water, and hygiene and health care are important components of poultry farm management. You may have seen TV news or read newspaper report about the bird flu virus which created a scare in the country and drastically affect egg and chicken consumption. Find out more about it and discuss whether the panic reaction was justified. How can we prevent the spread of the flu in case some chicken are infected? Animal breeding. Breeding of animal is an important aspect of animal husbandry. Animal breeding aims at increasing the yield of animal and improving the desirable qualities of the produce. For what kind of characters would we breed animal? Would the selection of characters differ with the choice of animals? What do we understand by the term breed? A group of animals related by descent and similar in most characters like general appearance, feature, size, configuration, etc. are said to belong to a breed. Find out the names of some common breeds of cattle and poultry in the farm of your area. What do we understand by the term breed? A group of animals related by descent and similar in most characters like general appearance, features, size, configuration, etc. are said to be. When breeding is between animals of the same breed, it is called inbreeding, while cross between different breeds are called outbreeding. Inbreeding. Inbreeding refers to the mating of more closely related individuals within the same breed for 4 or 6 generations. The breeding strategy is as follows. Superior male and superior female of the same breed are identified and mated in pair. The progeny obtained from such mating are evaluated and superior males and females among them are identified for further mating. A superior female in case of cattle is the cow or buffalo that produce more milk per lactation. On the other hand, a superior male is the bull which gives rise to superior progeny as compared to those of other males. Try to recollect the homozygous purloin developed by Mendel as discussed in chapter 5. A similar strategy is used for developing purloin in cattle as was used in case of peas. Inbreeding increases homozygosity. Thus, breeding is necessary if we want to evolve a purloin in any animal. Inbreeding exposes harmful recessive genes that are eliminated by selection. It also helps in accumulation of superior gene and elimination of less desirable gene. Therefore, this approach where there is selection at each step increases the productivity of inbreed population. However, continued inbreeding, especially close inbreeding, usually reduce fertility and even productivity. This is called inbreeding depression. Whenever this becomes a problem, selected animal of the breeding population should be mated with unrelated superior animals of the same breed. This usually helps restore fertility and yield. Outbreeding. Outbreeding is the breeding of the unrelated animals which may be between individuals of the same breed but having no common ancestor for 4 to 6 generations, outcross or between different breeds, crossbreeding or different species, interspecies hybridization. Outcross, this is the practice of mating of animals within the same breed but having no common ancestor on either side of their pedigree up to 4 to 6 generations. The offspring of such mating is known as an outcross. It is the best breeding method for animals that are below average in productivity in milk production, growth rate in beef cattle, etc. A single outcross often helps to overcome inbreeding depression. Crossbreeding. In this method, superior male of one breed are mated with superior females of another breed. Crossbreeding allows the desirable qualities of two different breeds to be combined. The progeny hybrid animals may themselves be used for commercial production. Alternatively, they may be subjected to some form of inbreeding and selection to develop new stable breeds that may be superior to the existing breeds. Many new animal breeds have been developed by this approach. Hizardale is a new breed of sheep developed in Punjab by crossing Bikaneri ewes and merino rams. Interspecific hybridization. In this method, male and female animals of two different related species are mated. In some cases, the progeny may combine desirable features of both the parents and may be of considerable economic value. Example, the mule. Do you know what cross lead to the production of the mule? Controlled breeding experiments are carried out using artificial insemination. The semen is collected from the male that is chosen as a parent and injected into the reproductive tract of the selected female by the breeder. The semen may be used immediately or can be frozen and used at a later date. It can also be transported in a frozen form to where the female is housed. In this way, desirable mating are carried. Artificial insemination helps us overcome several problems of normal mating. Can you discuss and list some of them? Often, the success rate of the cross mature male and female animal is fairly low even though artificial insemination is carried out. To improve chance of successful production of hybrids, other means are also used. Multiple ovulation embryo transfer technology (MOET) is one such program for herd improvement. In this method, a cow is administered hormone with SH-like activity to induce follicular maturation and super ovulation instead of one egg, which they normally yield per cycle. They produce 6 to 8 eggs. The animal is either mated with an elite bull or artificially inseminated. The fertilized egg at 8 to 32 cell stage are recovered non-surgically and transferred to surrogated mother. The genetic mother is available for another round of superior ovulation. This technology has been demonstrated for cattle, sheep, rabbits, buffalo, mares, etc. How high milk yielding breeds of female and high quality lean meat with less lipid. Meat yielding bulls have been bred successfully to increase herd size in a short time. Beekeeping. Beekeeping or apiculture is the maintenance of hives of honeybees for the production of honey. It has been an age-old cottage industry. Honey is a food of high nutritive value and also find use in the indigenous system of medicine. Honeybee also produce bee wax, which find many use in industry such as in the preparation of cosmetic and polish of various kind. The increased demand of honey has led to a large-scale beekeeping practice. It has become an established income-generating industry where practiced on a small or on a large scale. Beekeeping can be practiced in any area where there are sufficient bee pasture of some wild herb, fruits, orchids, fruit orchards and cultivated crops. There are several species of honeybee which can be reared. Of these, the most common species is Apis indica. Beehives can be kept in one's country yard, on the veranda of house, or even of the roof. Beekeeping is not labor intensive. Beekeeping, though relatively easy, does require some specialized knowledge, and there are several organizations in teach beekeeping. The following points are important for successful beekeeping. 1. Knowledge of nature and habits of bees. 2. Selection of suitable location for keeping the beehives. 3. Catching and hiving of swarms group of bees. 4. Management of beehives during different seasons. And 5. Handling and collection of honey and bee wax. Bees are the pollinator of many of our crop species such as sunflower, brassica, apple and pear. Keeping beehives in crops field during flowering period increases pollination efficiency and improves the yield, beneficial both from the point of view of crop yield and honey yield. Fisheries. Fishery is an industry devoted to catching, processing or selling of fish, shellfish or other aquatic animals. A large number of our population is dependent on fish product and other aquatic animals such as prawn, crabs, lobster, edible oyster, etc. for food. Some of the freshwater fishes which are very common include katla, rui and common carp. Some of the marine fishes that are eaten include hilsa, sardines, martins and pomfrets. Find out what fishes are commonly eaten in your area. Fisheries has an important place in India's economy. It provides income and employment to millions of fishermen and farmers, particularly in the coastal state. For many, it is the only source of their livelihood. In order to meet the increasing demand on fisheries, different techniques have been employed to increase production. For example, through aquaculture and pisciculture, we have been able to increase the production of aquatic plants and animals, both freshwater and marine. Find out the difference between the pisciculture and aquaculture. This has led to the development and flourishing of the fishery industry, and it has brought a lot of income to farmers in particular and the country in general. We now talk about Blue Revolution as being implemented along the same line as Green Revolution. Plant breeding. Traditional farming can only yield a limited biomass as food for humans and animals. Better management practice and increases in acreage can increase yield, but only to a limited extent. Plant breeding as a technology has helped increase yield to a very large extent. Who in India has not heard of Green Revolution, which was responsible for our country to not merely meet the national requirement in food production but also help us even to export it. Green Revolution was dependent to a large extent on plant breeding techniques for development of high-yielding and disease-resistant variety in wheat, rice, maize, etc. What is plant breeding? Plant breeding is the purposeful manipulation of plant species in order to create desired plant types that are better suited for cultivation, give better yield and are disease resistant. Conventional plant breeding has been practiced for thousands of years since the beginning of human civilization. Recorded evidence of plant breeding dates back to 9,000 to 11,000 years ago. Many present-day crops are the result of domestication in ancient times. Today, all our major food crops are derived from domesticated varieties. Classical plant breeding involves crossing or hybridization of pure line, followed by artificial selection to produce plants with desirable traits of higher yield, nutrition and resistance to disease. With advancement in genetics, molecular biology and tissue culture, plant breeding is now increasingly being carried out by using molecular genetic tools. If we were to list the traits or character that the breeders have tried to incorporate into crop plant, the first we would list would be increased crop yield and improved quality, increased tolerance to environmental stresses, salinity, extreme temperature, drought, resistance to pathogen, virus, fungi and bacteria, and increased tolerance to insect pests would be on our list too. Plant breeding programs are carried out in a systematic way worldwide in government institutions and commercial companies. The main steps in breeding a new genetic variety of a crop are: 1. Collection of variability. Genetic variability is the root of any breeding program. In many crops, pre-existing genetic variability is available from the relative of the crop. Collection and preservation of all the different wild varieties, species and relatives of the cultivated species, followed by their evolution for their characteristics, is a prerequisite for effective exploitation of natural genes available in the population. The entire collection of the plant seed having all the diverse allele for all genes in a given crop is called germplasm collection. 2. Evaluation and selection of parents. The germplasm is evaluated so as to identify plants with desirable combination of characters. The selected plants are multiplied and used in the process of hybridization. Pure lines are created wherever desirable and possible. 3. Cross hybridization among the selected parents. The desired character have very often to be combined from two different plants parent. For example, high protein quality of one parent may be needed to be combined with disease resistance from another parent. This is possible by cross hybridizing the two parents to produce hybrids that genetically combine the desired character in one plant. This is a very time consuming and tedious process since the pollen grain from a desirable plant chosen as male parents have to be collected and placed on the stigma of the flower selected as female parent. In chapter 2, details on how make crosses have been described. Also, it is not necessary that the hybrid do combine the desirable character. Usually only one in few hundreds to 1,000 process show the desirable combination for selection and testing of superior recombinant. This step consists of selecting among the progeny of the hybrids those plants that have the desired character combination. The selection process is crucial to the success of the breeding objective and require careful scientific evaluation of the progeny. These steps yield plants that are superior to both of the parents. Very often more than one superior progeny plant may become available. These are self-pollinated for several generations till they reach a state of uniformity, homozygosity, so that the character will not segregate in progeny. 5. Testing, release and commercialization of new cultivars. The newly selected lines are evaluated for their yield and other agronomic traits of quality, disease resistance, etc. This evaluation is done by growing these in the research field and recording their performance under ideal fertilizer application, irrigation and other crop management practice. The evaluation in research field is followed by testing the materials in farmers' field for at least 3 growing seasons at several locations in the country, representing all the agroclimatic zones where the crops is usually grown. The materials is evaluated in comparison to the best available local cultivar, a check or reference cultivar. India is mainly an agricultural country. Agriculture accounts for approximately 33% of India's GDP and employs nearly 62% of population. After India's independence, one of the main challenges facing country was that the producing enough food for increasing population. As only limited lands is fit for cultivation, India has to strive to increase yield per unit area from existing farmlands. The development of several high-yielding varieties of wheat and rice in the mid-1960s as a result of various plant breeding techniques led to a dramatic increase in food production in our country. This phase is often referred to as Green Revolution. Here represents some Indian hybrid crops of high-yielding varieties. Wheat and rice. During the period 1960 to 2000, wheat production increased from 11 million tons to 75 million tons, while rice production went up from 35 million tons to 89.5 million tons. This was due to the development of semi-dwarf varieties of wheat and rice. Nobel laureate Norman E. Borlaug at International Center for Wheat and Maize Improvement in Mexico developed semi-dwarf wheat. In 1963, several varieties such as Sonalika and Kollan Sona, which were high-yielding and disease-resistant, were introduced all over the wheat-growing belt of India. Semi-dwarf rice varieties were derived from IR8 developed at International Rice Research Institute IRRI, Philippines. The Taichung native one from Taiwan. The derivatives were introduced in 1966. Later, better yielding semi-dwarf varieties Joya and Rodna were developed in India. Sugarcane. Sakaram barberry was originally grown in North India but had poor sugar content and yield. Tropical cane growth in South India, Sakaram officinarum, had thicker stem and higher sugar content but did not grow well in North India. These two species were successfully crossed to create sugarcane varieties combining the desirable quality of high yield thick stem, high sugar, and ability to grow in sugarcane areas of North India. Millet, hybrid maize, jowar, and bajra have been successfully developed in India. Hybrid breeding have led to development of several high-yielding varieties resistant to water stress. Plant breeding for disease resistance. A wide range of fungal, bacterial, and viral pathogens affect the yield of cultivated crop species, especially in tropical climate. Crop losses can often be significant, up to 20 to 30%, or sometimes even total. In this situation, breeding and development of cultivars resistant to disease enhances food production. These also help reduce the dependence on use of fungicide and bactericide. Resistance of the host plant is the ability to prevent the pathogen from causing disease and is determined by the genetic constitution of the host plant. Before breeding is undertaken, it is important to know about the causative organism and the mode of transmission. Some of the diseases caused by fungi are rust, example brown rust of wheat, red rot of sugarcane, and lead blight of potato by bacteria, black rot of crucifers and by viruses, tobacco mosaic, turnip mosaic, etc. Method of breeding for disease resistance. Breeding is carried out by the conventional breeding techniques described earlier or by mutation breeding. The conventional method of breeding for disease resistance is that of hybridization and selection. Its steps are essentially identical to those for breeding for any other agronomic character, such as high yield. The very sequential steps are screening germplasm for resistance sources, hybridization of selected parents, selection and evaluation of hybrids, and testing and release of new varieties. Some crop varieties breed by hybridization and selection for disease resistance to fungi, bacteria, and viral disease are released. Conventional breeding is often constrained by availability of limited number of disease resistance genes that are present and identified in various crop variety or wild relatives. Inducing mutation in plants through diverse means and then screening the plant materials for resistance sometimes lead to desirable genes being identified. Plant having this desirable character can then be either multiplied directly or can be used in breeding. Other breeding methods that are used are selection among somaclonal variant and genetic engineering. Mutation is the process by which genetic variations are created through changes in base sequence within gene, resulting in the creation of new character or trait not found in parental type. It is possible to induce mutation artificially through use of chemical or radiation like gamma radiation and selection, and using plants that have the desirable character as a source in breeding. This process is called mutation breeding. In mung bean, resistance to yellow mosaic virus and powdery mildew were induced by mutation. Several wild relatives of different cultivated species of plant have been shown to have certain resistance character but have very low yield. Hence there is a need to introduce the resistance resistant gene into high-yielding cultivated varieties. Resistance to yellow mosaic virus in bhindi, apple, mosas, esulentus was transferred from a wild species and resulted in a new variety of A. esulentus called Parbhani Kranti. All the above examples involve source of resistance genes that are in the same crop species, which has to be bred for disease resistance or in a related wild species. Transfer of resistance genes is achieved by sexual hybridization between the target and the source plant followed by selection. Plant breeding for developing resistance to insect pests. Another major cause for large-scale destruction of crop plant and crop produce is insect and pest infestation. Insect resistance in host crop plant may be due to morphological, biochemical or physiological characteristics. Hairy leaves in several plants are associated with resistance to insect pests. Example, resistance to jacid in cotton and cereal leaf beetle in wheat. In wheat, solid stem lead to non-preference by the stem soft leaf and smooth leaf, and nectarless cotton varieties do not attract bollworm. High aspartic acid, low nitrogen and sugar content in maize lead to resistance to maize stem borage. Breeding method for insect pest resistance involve the same steps as those for any other agronomic trait such as yield or quality and are as discussed earlier. Sources of resistant genes may be cultivated varieties, germplasm, collection of the crop or wild relatives. Some released crop varieties bred by hybridization and selection for insect pest resistance are given in table. Plant breeding for improved food quality. More than 840 million people in the world do not have adequate food to meet their daily food and nutritional requirement. A far greater number, 3 billion people, suffer from micronutrient, protein and vitamin deficiencies or hidden hunger because they cannot afford to buy enough fruits, vegetables, legumes, fish and meat. Diet lacking essential micronutrients, particularly iron, vitamin A, iodine and zinc, increases the risk of disease, reduce lifespan and reduce mental abilities. Biofortification. Breeding crop with higher level of vitamins and minerals or higher protein and healthier fat is the most practical means to improve public health. Breeding for improved nutritional quality is undertaken with the objective of improving on protein content and quality, 2 oil content and quality, 3 vitamin content and 4 micronutrient and mineral content. In 2000, maize hybrids that had twice the amount of amino acid, lysine and tryptophan compared to existing maize hybrid were developed. Wheat variety LR66 having a high protein content has been used as a donor for improving cultivated wheat. It has been possible to leave an iron fortified rice variety containing over 5 times as much iron as in commonly consumed varieties. The Indian Agriculture Research Institute New Delhi has also released several vegetable crops that are rich in vitamins and minerals. Example, vitamin A enriched carrot, spinach, pumpkin. Vitamin C enriched bitter gourd, bathua, mustard, tomato. Iron and calcium enriched spinach and bathua, and protein enriched beans, broad, lab, French and garden peas. Single cell protein (SCP). Conventional agricultural production of cereal, pulses, vegetables, fruits, etc. may not be able to meet the demand of food at the rate which human and animal population is increasing. The shift from grain to meat diet also create more demand for cereal, as it takes 3 to 10 kg of grain to produce 1 kg of meat by animal farming. Can you explain this statement in the light of your knowledge of food chain? More than 25% of the human population is suffering from hunger and malnutrition. One of the alternate source of protein for animal and human nutrition is single cell protein. Microbes are being grown on an industrial scale as source of good protein. Blue-green algae like spirulina can be grown easily on materials like wastewater from potato processing plant containing starch, straw, molasses, animal manure and even sewage to produce large quantities and can serve as food rich in protein, minerals, fat, carbohydrate and vitamins. Incidentally, such utilization also reduces environmental pollution. Certain bacteria like species like Methylophilus, Methylostrophus, because of its high rate of biomass production and growth, can be expected to produce 25 tons of protein. The fact that edible mushrooms are eaten by many people and large-scale mushroom culture is a growing industry make it believed that microscopic fungi too would become acceptable as food. Tissue culture. As traditional breeding techniques fail to keep pace with demand and to provide sufficiently fast and efficient system for crop movement, another technology called tissue culture got developed. What does tissue culture mean? It was learned by scientists during 1950s what whole plant could be regenerated from a plant, that is any part of the plant taken out and grown in a test tube under sterilized conditions in special nutrient media. This capacity to generate a whole plant from any cell or explant is called Potency. You will learn how to accomplish this in higher classes. It is important to stress here that the nutrient medium must provide a carbon source such as sucrose and also inorganic salt, vitamin, amino acid and growth regulator like auxin, cytokinin, etc. By application of this method, it is possible to achieve propagation of a large number of plants in a very short duration. This method of producing thousands of plants through tissue culture is called micropropagation. Each of these plants will be genetically identical to the original plant from which they were grown, that is, they are soma clones. Many important food plants like tomato, banana, apple, etc. have been produced on commercial scale using this method. Try to visit a tissue culture laboratory with your teacher to better understand and appreciate the process. Another important application of method is the recovery of healthy plant from diseased plant. Even if the plant is infected with virus, the meristem, apical and auxiliary is free of virus. Hence, one can remove the meristem and grow it in vitro to obtain virus-free plant. Scientists have succeeded in culturing meristem of banana, sugarcane, potato, etc. Scientists have even isolated single cell from plant and after digesting their cell wall have been able to isolate naked protoplasm surrounding by plasma membrane. Isolated protoplast from two different varieties of plant, each having a desirable character, can be fused to get hybrid protoplast which can be further grown to form a new plant. These hybrids are called somatic hybrid, while the process is called somatic hybridization. Imagine the situation when a protoplast of tomato is fused with that of potato and then they are grown to form new hybrid plants combining tomato and potato of characteristics. Well, this has been achieved resulting in formation of tomato. Unfortunately, this plant did not have all the desired combination of characteristics for its commercial utility.